now we will have the talk of Dr. Katya Romero Soler. Katya is a brand new researcher at the University of the Autonomous University of Querétaro in Mexico. So she's eager to get collaborations in Bromeliaceae. And she will be presenting the diversification of Hechtioideae Bromeliaceae in the dry forest of North and Central America.
Thank you, Carolina, for the introduction. So Caro was talking us about this huge Bromeliad group. And now I want to talk you about this spiny Bromeliad group. In the next slides, I want to share with you what we know about this subfamily, what we are doing now, and what we want to do in the in the near future. So what is Hechtioideae? This subfamily of bromeliads is found growing from southern United States to northern Nicaragua. However, most of the species are growing in Mexico, around 96% of the 100 species recognized. In fact, most of the species are endemic to Mexico. And we now recognize 3 genera. Species in this group are terrestrial plants and succulents. And typically, we can find them growing in more dry forests. Most of the species grow in this type of biome. But as also, we can find some species growing in deserts and xeric shrublands. And a few species some species can grow in pine and oak forests, and some, really a few species in more moist forests. But we need to check that really. Morphologically, this group is a really painful group because here, we can find succulent plants, but also they have really, really spiny leaves. And it's really difficult to collect. And maybe, I don't know if you have known some species of Hechtia, or maybe other species like Dickia or things like that I can imagine that also have really painful spines. And Hechtioideae is also interesting because the species have unisexual flowers and are dioecious plants. I think this is really interesting in this group, and we don't know much about this. But there is a species, which is Hechtia iorum, that also have unisexual flowers, but you can find female and male flowers in the same individual, also perfect flowers. But this species is only known from the type locality, and we really don't know nothing about that. About the fruits, are capsular, as most of the species in Bromeliaceae, they have a small wings or be completely naked, and they have low vigility. In fact, the amount of endemicity that we can found in this group is associated to this low capacity of seed dispersion. What we know about the phylogenetic relationships in this group, this is a phylogenetic tree, and in the first phylogenetic tree that we published in 2018, it was based in 2 plastid loci and intergenic spacer and fragments of the YCF1 gene. And here we include also a nuclear gene, the PRK, because we didn't have success in amplifying other Bromeliad loci like 5C or ETS. So the only that we can use was that. And we also include 5 morphological characters in that moment. And as taxon sampling, we included 50 species of the subfamily. We found that the subfamily was monophyletic, as was previously were known. And additionally, we identified 3 main clades: a small clade that includes Central American species, other clade that include only Mexican species, and a big clade that includes most of the species of these groups. So in that moment, we did taxonomic changes based on the phylogenetic hypothesis that we have, based on the morphology that we understand in this group, and also based on the geographical distributions. So I want to present you these 3 genera. The first one is Bakerantha, which is the one that is here, includes 5 species distributed in central eastern region of Mexico. And these plants are really nice because, well, I worked with this group in my PhD. And these plants grow on rocky soils and vertical walls. In fact, I think it's difficult to see, but if you, oops, sorry. Here you can see like grape grass plants, and those are Bakerantha species, and they're really nice plants. And in the case of these species, they don't have spines, so that is the funny part of this genus. But they have the margins are serrated, so they could be also painful in that part. And here we can find flowers that are in really beautiful inflorescence with a lot of small flowers. With the flowers have pedicels, really thin pedicels, and the petals can be white, lilac, pink. And the other genus includes 3 species, which are Central American species, and they grow in Belize, Guatemala to Nicaragua. And the species in this group, 2 of them grows in pine and oak forest, and in one of them in dry forest. Here you can see an example how looks one of the species in this group. Also, they grow as terrestrial as succulent. They have small spines, and these are more soft than those on Hechtia. And we distinguish this group by the sessile flowers and the ovary, which is mostly inferior, and have white petals, but also the apex of the petals could be red. And then is Hechtia, which is the genus that includes most of the species, 92 species, and is really widely distributed. Here we found within the interior of Hechtia, which is all these species that are here, we found several clades. One of these clades is the complex Hechtia glomerata, which includes lateral inflorescence. I remember the talk of Rachel talking about these Hechtia species which produce the same rosette can produce multiple inflorescence. So that we can found that in this in the Hechtioideae complex. So the species here have white petals, have a lot of indument even in the fruits, and there is only one species that have pink flowers. There is other small clade, which we call clade E, that include the 2 species with complete inferior ovary. And there is other clade that we call clade F, which include the remaining species of Hechtia, which is really morphologically diverse. And what we can see here is that the clades that we identify with this phylogenetic tree have more geographical similarity than morphological similarity. Then 2 years ago, we published other phylogenetic hypotheses of this group using the same loci, but here we expand our taxonomic sampling. But in this case, the objective was not to propose other phylogenetic hypotheses. In this case, we want to know about the phylogenetic diversity of the species among the distribution. But in this moment, we did phylogenetic dating, and we found that most of the species within Hechtia arise at least 3.5 million years ago. But we also found incongruence between the relationships in this group. So having these two phylogenetic hypotheses with low resolution, low statistical support, and incongruence, we decided to try to prove other kind of data. So that's where we are now. So we were exploring the plastome data. We did, in this case, whole genome sequencing. So all the genome, the effort was put in sequence plastomes, because that was the object in that moment. And we newly sequenced 19 species of Hechtioideae, and we found that the plastomes have a size of 155 to 165 kilobytes. And we also found that the gene order and content is generally conserved. Just remember the numbers that Carolina gave us in her talk at the similar in Hechtioideae. But here we found some interesting things. We found some structural rearrangements. We found inversions in only one of the species that we include in this study, and was related to an inversion of 4 genes in one of the species with inferior ovary. We also found expansions, one expansion in this same species that includes an expansion of the inverted repeat and a reduction of the small single copy. And there are 4 genes that are duplicated now in this species. We also found something that I think is really interesting. We need to study in the future, that is loss and potential pseudogenization of the genes in the NDH family. So in the case of this gene pseudogenization, we found that some genes have punctual mutations and some base insertions and deletions, and in some cases, most of the gene is lost. We can see maybe a gene that have 100 pair bases. We just have in this case 5 pair bases. So most of the gene has gone. So we check that, verified each of these changes by assessing the coverage of the data. In this case, we have really good coverage. So now we need to know where are these genes, right? If they are really lost, or maybe we need to explore the nuclear and the mitochondrial genome to see what is happening, as has been seen in other plants, right? And also it's interesting because most of these changes are in the clade F, so the more derivative space. Oh, we also made a phylogenetic reconstruction using these plastome sequences. Here we found that Mesoamerantha, the clade that includes the Central American species, is sister to Hechtia and Bakerantha. And we also found that using all this data allow us to obtain a better and a stronger node support in the phylogenetic relationships in this subfamily. We also did this primer or phylogenetic informatives analysis to see how was to identify possible markers for future studies in this subfamily. And we found that the previous markers that we were using, not just for phylogenetic reconstruction, also for phylogeographical studies, were not among the most informative loci. So only one of the genes that we used previously, that was the YCF1 gene, is the most informative. And we also compared the most informative loci between Hechtioideae and Phylogenetics, and only a few loci were shared. So in this case, we think that it's better to use maybe a specific marker. Well, it depends that what we want to know, right? And now we have new data. We have been using this same high-seq strategy that Carolina talked us in her talk. So I don't going to explain you about that because she already mentioned that. And here we use our taxon sampling includes 72 Hechtioideae species, which represent the 72% of the species that are recognized now. No? So to show you how we think is the power of using plastomes in this kind of Bromeliad group, or especially in this group, I also going to show you a phylogenetic tree using the traditional markers that we have been using in phylogenetics. So here in the left, yes, that is the left, right? Left, right. Okay, left. It's based on 2 plastid loci that we were implementing in Hechtioideae phylogenetics. While in the right, you can see the tree based on the complete plastomes. The red dots show the node support. That is 91 to 100, and the green dots there are just a few, represents not support about 81%. And those nodes that are not, don't have a dot, so don't have a node support below 18. As you can see, there are a lot well-supported clades compared with the phylogenetic hypothesis that we have using Sanger sequencing. So we think this is really a good opportunity to have a really robust phylogenetic hypothesis in this Bromelia group. So, well, as you can see here, again, we recovered that Mesoamerantha, the species from Central America, are sister to or other Bakerantha and Hechtia. So also plastome sequences solved several recalcitrant relationships in Hechtioideae, thinking about the phylogenetic relationships in this clade F, that are these species here and here. And we here we can see the names of the clades and geographical regions identified in the first phylogenetic hypothesis based on Sanger sequencing. Most of them are conserved with some changes in species composition, but in particular, every clade that we recognize with Sanger sequencing, we could recover these clades here, but with better support and resolution within that clades. We couldn't recover the clade E, sorry, that then including this species with the inferior ovary. And in that sense, I want to just to comment you that in this phylogenetic hypothesis that we used, we did using expanded taxon sampling that the main in that work was to do a phylogenetic diversity analysis. There we also did an estimation of ancestral areas, and it was proposed that a pattern where dispersion to new geographical areas and at subsequent species diversification characterize these groups. So we want to redo that analysis now that we have this more strong phylogenetic hypothesis. We also did a dating phylogenetic analysis for this group. And you can see here, the branches are too small in this group. As you can see, well, I think you can see the branches. So here we found that the stem age of the subfamily is 21 million years, and the crown age is about 14 million years. That is a little oldest than the previous estimations for this group. We found that the crown age of the genera it's about 9 to 7 million years ago. However, this is really, well, for me it's really interesting because in Hechtia, in core Hechtia, as I put there, most of the species diverged in the last 2 million years ago. So this suggests a recent and rapid diversification in this group. So it's an interesting group thinking about this morphological complexity that I was telling you in the first slides. So what's next? Well, we want to know where did the diversification of Hechtioideae occur? Was it associated with any morphological or ecological trait? And have there been significant changes in the rate of diversification of this interesting group? And as well, as you can see, this is a team effort. And this is just part of the persons that have been working for a long time in this group. And I just want to recognize their work. And thanks also the people that gives us the money. And a special thanks to the Bromeliaceae Society International, which help us with the travel expenses. And thank you to you for listening to me.
Thank you, Katya.
Mhm.
Any questions?
wonderful talk and really exciting results. I was just curious, and I would never hold you to this. Would you see any reason to elevate any of the clades within Hechtia to the genus level?
Sorry, again?
Oh, do you see any, now that you have a well-resolved phylogeny within Hechtia, do you see any need to elevate any clades to a genus?
I don't think so.
Okay.
No, because there is a lot of yes, there is a lot of morphological variation there, but I don't think that we need to create other genus, because in the case of Bakerantha, for example, because it's really different. You see the plants there, you see the flowers, you see the inflorescence. Also in the case of the Mesoamerantha, but within Hechtia, I think there are a lot of shared characteristics, but I think Hechtia should be.
I like it.
Hechtia.
Great answer. Thank you.
So yes, great results, and think how far you've gone in just a few years from almost no resolution to almost complete resolution. I guess my only question had to do with the dating. So when we did the cross monocots analysis using 20 fossils and calibration points, or when we did the Bromeliad calibration, those stem ages are not possible. What are you seeing as the age of bromeliads if you're looking at the stem age of Hechtioideae it being 23 million years? Again, we were finding 20 million years for the crown age of the family.
Yeah. Here we use for calibration, well, we use secondary calibrations, and we use the calibration based in the angiosperm phylogeny of Ramirez Barahona, that is along the angiosperm. And here we didn't use like punctual calibrations to did the so we left all the three.
So just with respect, I think we had
Yeah.
a greater density
Yeah.
of calibration points
Yeah.
than that study, and you make your judgment, but
Yeah,
I would
it's
advise
still
that you probably have too old ages.
Yeah. Yeah. Mm.
We have time for one more question.
Thank you. Very nice talk, Katya. Are there any C3 plants in that clade of Hechtia? And if there are, did you see things in the chloroplast that are different?
C3.
C3 versus CAM?
Okay. Well, all the species are CAM, but if I remember the Crane paper, there are these species from Bakerantha which grow in like near the boundaries of moist forests, have a low index of CAM, so they are like in the limits to C3. So we didn't check for things like that in our chloroplast.
Thank you, Katya.
Thank you.